Okay, I'm sorry. The splitting of these branches have found to destroy the people's properties and have caused damage to their cars or even their houses, causing huge economic loss. Okay. Again, several escape populations of Iris scalariana also have sharp thorns in them, which make it difficult for the individuals in the forest to remove those. These sharp thorns even have the potential to puncture the tires of vehicles, and there have been several instances where the tires of vehicles were found to be punctured by those tires, again causing huge economic loss. Though the Galeriana flowers are very visually appealing and beautiful, the smell is awful. Trust me, the smell is really awful. People have described the smell as a rotten egg or rotten fish. Some have even described the smell as semen or urine or even animal wastage. So say no to Paris Galeriana. So for my thesis, I worked on two of the projects related to this invasive tree. The first project was molecular detection of Pyrus scalariana using loop-mediated isothermal amplification. And the second project was assessment of genetic diversity of Pyrus scalariana using mitochondrial microsatellites. Talking a little background for the first project, this tree has been reused as a rootstock as well as for landscaping purpose for a very long period of time. However, several states are restricting the sales of Iris scalariana, and in fact, some states like Ohio, Pennsylvania, and South Carolina have already banned the sales of Pyrus scalariana, and some states are planning to ban its sales. Since PC has been used as rootstocks alongside other closely related species such as Pyrus communis, Pyrus petulifolia, Pyrus pirifolia, it is at first important to be able to differentiate the rootstock of Pyrus scalariana from the rootstock of other three species. So to aid the state in enforcing regulatory measures is a step to limit the ongoing spread of Pyrus scalariana. A field-based rapid detection tool is required that could reliably detect Pyrus scalariana DNA. However, at present, we don't have any field-based detection tool that can differentiate the rootstocks of Pyrus scalariana from the rootstocks of other three species, namely Pyrus communis, petulifolia, and pirifolia. Therefore, the broad objective of our research was to develop a rapid field-based detection tool for Pyrus scalariana that could reliably detect its DNA. So to achieve this broad objective, we had certain specific objectives set. The first objective was to identify and characterize candidate regions of Pyrus scalariana through comparative genomics of plastidial genome, mitochondrial genome, and nuclear genome across Pyrus species. The second objective was to evaluate those candidate regions and test whether they are suitable in the development of LAMP protocol or not. And finally, the third objective was to develop an optimized LAMP assay for the field detection of Pyrus scalariana. Before talking about how each of those objectives were made, let me talk a little bit about LAMP. LAMP is an isothermal amplification technique in which the amplification and detection takes place in a single step by incubating all the necessary reaction mixtures at constant temperature. This molecular detection tool is widely used throughout the tree of life and is a very well-established method. Unlike conventional PCR detection tools, no expensive thermocyclers are required for LAMP, which makes it an economical method. Also, its rapidity, high specificity, and high sensitivity because of use of multiple primers make it different from conventional PCR techniques. LAMP basically works with 4 to 6 primers. 4 primers are the core primers, which include forward and backward outer primers, F3B3, and forward and backward inner primers, which are FIP and BIP. These are the prerequisites for LAMP, and LAMP does not work in absence of any of these 4 primers. However, there are additional 2 primers, which are optional, which are the loop forward and loop backward primers. LAMP works even in the absence of these primers. However, with the addition of just one or both of the loop primers, the speed of LAMP reaction increases, which enhances the specificity of the LAMP reaction as well. Another good thing or advantage of using this tool as a detection tool is that it is very easy to interpret the results of LAMP. So no prior knowledge in molecular detection is needed just to interpret the LAMP results. As you can see in these pictures as well, the picture on the top shows the LAMP results just by the change in turbidity or by the formation of precipitation. The other picture shows LAMP amplification just by the change in color from pink to yellow. So the simplicity of LAMP makes it feasible for the actual deployment of this tool in the field. So to achieve our first objective, we at first tried identifying the candidate regions in plastidial and mitochondrial genome across Pyrus species. This figure shows the MVista view where the mitochondrial genome of Pyrus petulifolia, Pyrus pyrifolia, and Pyrus communis have been aligned, keeping the mitochondrial genome of Pyrus scalariana as the baseline or the reference. All the genomes used as input in MVista were retrieved from NCBI, and these regions within these rectangular boxes represent the regions of low sequence similarity in comparison to Pyrus scalariana. So by this approach, we were able to retrieve 19 potential targets from a plastidial genome and 16 potential targets from the mitochondrial genome. Out of all those potential targets, 9 targets from plastidial and 13 from mitochondrial genome were able to yield primers under default parameters of NEB LAMP primer design tool. So from this approach, we're able to get 9 plastidial and 13 mitochondrial primer sets to test further. So after identifying those candidate regions and developing primers, the F3B3 outer primers from the plastidial genome were evaluated using PCR. In this PCR picture, we can see the 300 BP was the expected product size. The numeric figure followed by the PCR number indicates the approximate position of each primer sets in the plastidial genome of Pyrus scalariana. For testing, we used 4 different samples of Pyrus scalariana as well as the samples of other Pyrus species and outgroup species. PCR results showed positive amplification for almost all of the samples tested. The same evaluation was done for the mitochondrial F3B3 primers using the same sets of samples and the same PCR condition. After that, the PCR products were sent for Sanger sequencing, and the Sanger sequenced results were assembled and aligned and visualized in BioEdit. This is one of the candidate loci that was used for downstream analysis. The sequence here on the top represents the reference genome of Pyrus scalariana, against which all other F3B3 sequences were aligned. These 4 sequences represented the F3B3 sequence of 4 samples of Pyrus scalariana, and the next box containing 3 different sequences represented the sequences of non-target species, that is Pyrus communis, Pyrus pyrifolia, and Pyrus betulifolia. As you can see in the second box, there are nucleotide mismatches or polymorphism. So the reason somewhere around this region could be in the plastidial genome of Pyrus scalariana could be the potential candidate region, which could be specific to PC and differentiate it from other non-target species. So after evaluating the primers with PCR and Sanger sequencing, we tested those primers under LAMP. Basically, LAMP primer design tool yields different potential sets of primers. So initially, we selected only those primers that had either one or both of the loop primers in them. Because as I already told, the addition of loop primers enhances the specificity and speed of the LAMP reaction. We use Wormstead colorimetric LAMP master mix for LAMP. So all 9 plastidial and 13 mitochondrial primer sets were tested at 65 degrees centigrade with Pyrus scalariana and other species for different periods of time. The positive amplification was seen as early as 20 minutes with 5 ng per microliters of DNA. As you can see in this picture as well, the yellow color represents the positive amplification, whereas the pink represents the negative or no amplification. However, with this test, we're not able to get any primer set that was specific to Pyrus scalariana, meaning all primer sets all showed positive amplification for other non-target species as well. So after that, I tested for the primer sets without loop primers. There were altogether 15 sets of mitochondrial primers that had no loop primers. Again, unfortunately, no primer sets within these groups were specific to PC, and which could differentiate it from all non-target species. So again, I tried another approach to design LAMP primers utilizing the Sanger sequencing results. Initially, we had utilized online available genomes to screen for the candidate regions and develop LAMP primers out of those regions. But this time, we utilized experimentally verified F3B3 sequences of Pyrus scalariana and developed LAMP primers. There were altogether 4 sets of plastidial primers and 27 sets of mitochondrial primers. However, again, we were not able to get any primer specific to Pyrus scalariana that could differentiate it from other non-target species. After several unsuccessful attempts, we tried for another approach, which was GLPD. GLPD is a whole genome-based LAMP primer designing tool that accepts the genomes as input. GLPD accepts target groups and background groups as its input. In our case, target group included the genomes of Pyrus scalariana and background group included the genomes of 3 non-target species. The available whole genomes, plastidial genomes, and mitochondrial genomes of different species were retrieved from online databases, and GLPD was run separately on each of those assemblies to develop the LAMP primers. This program GLPD has an optional flag for whether to include the loop primers or not. So we tried for both of the options with and without loop primers. However, GLPD did not yield any primers for plastidial genome and whole genomes, but it yielded 5 sets of primers for mitochondrial genome. Out of all those 5 primers, the primer set MT26K. So 26K is the approximate position of the primer set within the mitochondrial genome of Pyrus scalariana. This particular set could at least differentiate Pyrus scalariana from both Pyrus betulifolia and Pyrus pyrifolia. However, it showed positive amplification for Pyrus communis. In this picture also, yellow color represents the positive amplification, whereas pink is the negative. So again, even with GLPD primers, we're not able to get a specific primer that could differentiate PC from all 3 non-target species. So again, with all those unsuccessful attempts, we again tried for finding LAMP primers utilizing OrthoFinder. For that, the orthologs assemblies of all Pyrus species were inferred using OrthoFinder. Then a custom Python script was used to extract the genes orthologous in 90% of the taxa. And then we utilized StatX to calculate the nucleotide similarity scores for each of the positions of interleaved sequences. After that, we again used a custom Python script to average these similarity scores. And the genes with the lowest average similarity scores were manually searched for nucleotide polymorphism in CView. The genes that had at least 3 nucleotide polymorphism specific to Pyrus scalariana were selected and extracted, and NCBI blast search was done for those genes to ensure that those genes were specific to Pyrus scalariana. Finally, by utilizing those candidate genes, LAMP primers were designed using LAMP primer, NEB LAMP primer design tool. So for identifying this candidate region, Jackson helped us with all this, so I can thank enough to Jackson. Utilizing OrthoFinder, we got 2 sets of LAMP primers. The inner and outer primers were the same for both sets of primers, the only difference being the loop backward primer. No convenient loop forward primer was generated for this region. The primer set, which is referred to as MYB1, has loop backward primer 1, and the primer set, which is referred to as MYB2, has LB2 as the loop backward primer. So in the preliminary test, while testing using the MYB1 primer set, we were able to get positive amplification just for Pyrus scalariana DNA. And there were no negative amplification for other 3 non-target species when tested under LAMP for 30 minutes. So when the same primer set was tested without the loop primer, we could not get positive amplification for even 1 hour assay. This shows how important loop primers can be to speed up the reaction as well, enhance the specificity of the reaction. So after preliminary test for confirming the specificity, I tested those primers using collection of DNA from Pyrus scalariana and non-target Pyrus species. As you can see in this table, out of Total 95 samples of Pyrus scalariana tested. 75 samples showed positive results within 30 minutes. However, there were five samples for which the result was ambiguous. The color of the reaction was neither yellow nor pink. It was kind of oranges. So when I increased the reaction time to 40 minutes, all of the ambiguous results of Paris Galeriana changed to positive. So for 40 minutes reaction, there was positive amplification for 80 of the samples out of 95. However, there were still 15 samples that had a negative result. This false negative could be due to the suboptimal quality of DNA that we used for this study. The Paris Galeriana samples, the most of the samples were from the major parts of the historical herbarium specimens. So the old DNA or having not so good quality could be one of the reasons for this false negative result. In case of other Pyrus species, we got no positive results for Pyrus communis and Pyrus perifolia. However, we had four false positive results for Pyrus petulifolia. This false positive result could have been due to cross-contamination while I was performing the experiments in the lab, or could have been due to the mislabeling of the samples while I was extracting DNA or labeling the tubes, or when thousands of samples are collected in the field, there might have been mishandling or a mislabeling of samples, potentially could be the reason for false positive results of Pyrus betulifolia. So after that, we tested for the sensitivity of LAMP assay. For that, I at first utilized one of the mitochondrial primer sets that had all 6 LAMP primers. For sensitivity analysis, I did 10-fold serial dilution starting from 5 NZ and going all the way down to 5 FZ. Um, uh, LAMP was run for a different period of time from 15 minutes to 30 minutes. And here we were able to get positive amplification within 20 minutes using 5 NZ per microliter of DNA. Again, here pink is negative and yellow is the positive. The detection limit for 30-minute reaction was just 0.5 pz, which is approximately 10 copies of the target region. So again, I tested for the sensitivity of LAMP assay using the actual primer that was found to be specific to Pyrus scalariana, but it had only 5 LAMP primers. I did the same thing. In this case, the positive amplification was observed within 30 minutes using 5 NZ per microliters of DNA. Now to test whether it is applicable to directly extract DNA or not in the field, I tried this approach. For this, I took 2 ml microcentrifuge tube and added 10 to 20 mg of plant tissues. The tissues included leaves, flowers, bars, and roots of Pyrus scalariana, which I collected from one of the cultivars that we have maintained in our bow house. So in a 2 ml microcentrifuge tube, I added 10 to 20 mg of the plant tissue and also added 100 to 300 microliters of the elution buffer. I just use a disposable plastic pestle just to gently grind the tissue and suspend the DNA in the solution. And then I use this solution containing DNA under LAMP to test whether we get any positive results. And we were able to get a positive amplification within 20 minutes. However, there was no positive amplification without dilution. Could be due to high concentration or of DNA, or could be due to the high viscosity of DNA without dilution, by high viscosity of solution without dilution, or due to the presence of PCR inhibitors prior to dilution. Uh, so after testing all those primer sets, we were able to get MYB1 primer set as a specific primer that could differentiate Pyrus calariana DNA from the DNA of other 3 non-target species. And finally, we were able to develop LAMP assay. For that, we just need to add a little amount of tissue in a 2 ml tube, add the buffer, just grind it and use it in a portable mini PCR machine to see the LAMP reaction. So this was the machine I used for this LAMP reaction. It is very small, portable. It is as light as 500 grams. It is battery power and it can be taken to wherever we want in the field. It can be connected to your smartphone or your computer and regulate the LAMP reactions through an app. So LAMP is very simple method which can be done very easily in actual field. In near future, we are planning to do ring test where we will be requesting different laboratories to perform the lamp assay. We'll be providing them with all the necessary ingredients, the lamp mix, primers, the samples, and everything. And they will perform the lamp assay just to ensure that this assay works. So all in all, lamp assay for invasive plant species have not been described in the literature before. So we were able to develop lamp assay that could differentiate Pyrus caladiana from other three non-target species. The major problem that we faced during this project was the primer design. It was really tough to design the specific primers for Pyrus caladiana. So the major significance or implications of this lamp tool is its utilization in enforcing the regulatory measures by the states. At this stage, where states are banning the cells of Pyrus caladiana, this LAMP tool is a pivotal advancement in managing the ongoing spread of Pyrus caladiana. So going to the second project, which was assessment of genetic diversity of Pyrus caladiana using mitochondrial microsatellites. The genetic diversity and population structure study is very much important to formulate effective management strategies for any invasive species. In case of Pyrus caladiana, previous studies have already utilized nuclear short sequence repeats to study the genetic diversity. The plastidial genome of Pyrus caladiana has already been developed, but no SSRs were found within the plastidial genomes. However, neither the mitochondrial genome nor the mitochondrial SSRs have been developed previously. So developing the mitochondrial genome itself provides a valuable addition to the existing genomic resources for this species. Also, utilizing mitochondrial SSRs for genetic diversity indices provides insights into the genetic variability between the native species and the US released commercial cultivars and the escapees. The presence or absence of agreements between nuclear and mitochondrial signals will provide information about the intensity of gene flow or recombination. Therefore, the objectives of the second project were to develop mitochondrial genome of Pyrus caladiana. The second objective was to develop and evaluate novel mitochondrial microsatellites. And finally, the third objective was to assess genetic diversity of Asian populations, naturalized US escaped populations, and the commercial cultivars utilizing mitochondrial SSRs based genotyping. To develop the mitochondrial genome, we utilized whole genome sequencing data of Pyrus caladiana, and we used NovoRap for assembling the genome. Out of several possible genome architectures, 6 were deemed to be circular or complete by the NovoRap. We used those 6 genome architectures and aligned them in MVista, keeping the mitochondrial genome of Pyrus petulifolia as the reference, because Pyrus petulifolia has the largest mitochondrial genome within the Pyrus species. So after aligning in MVista, we identified the potential gaps. Those observed gaps in the assemblies indicated potential errors or variations in the assemblies. So we developed missing point primers for those identified gaps and tested in using PCR. The solution that exhibited amplification of all those investigated gaps in the final PCR was selected as the correct assembly. Finally, the assembly was polished and annotated. After developing the mitochondrial genome, we used UST-SSR finder to identify the SSRs if present any. So UST-SSR finder was able to identify 5 mitochondrial SSRs from the mitochondrial genome. So this is the circular mitochondrial genome of Pyrus caladiana. This genome contains 15 core genes and contains all the typical genes present within the mitochondrial genomes of Pyrus species. And these are the positions of the 5 mitochondrial SSRs that we developed in this study. After developing mitochondrial genome, we generated phylogenetic tree using different mitochondrial genomes of species of rosaceae. The Pyrus species here highlighted with dark green color are the Asian species of Pyrus. The one in the blue is the European Pyrus species, and the ones in dark red color are the cultivars, which are developed by crossing 2 different Pyrus species. For evaluating mitochondrial microsatellites, we did PCR in 72 Asian populations, 67 US cultivars representing 12 unique cultivars, 90 populations from Tennessee skip populations, and 90 samples from southern skip populations. Southern skip populations included the populations from Georgia and South Carolina. In addition, we also evaluated 89 specimens of 16 virus species, 2 specimens of Malus rocki to evaluate the cross amplification potential of those developed mitochondrial SSRs. After doing PCR, the PCR products were um, visualized, sized, and cleaned using KXL capillary electrophoresis system. After that, the raw alleles were beamed using FlexBean, and finally, the clone correction was done using popper package in R. So this table represents the genetic diversity indices of Pyrus scalariana based on different populations. No CC is the sample size before clone correction, and CC is the sample size after clone correction. So there is a reduction in the sample size after clone correction, which indicates the presence of identical replicates in each of the populations tested. The missing data was found only in Asian populations. These missing data could be again linked to the suboptimal quality of DNA that we used for this study, as I have already mentioned while discussing about in the first project. So this missing data could be due to the low quality of DNA rather than being the null alleles. The highest number of alleles and high gene diversity was found to be in US commercial cultivars. The highest value of gene diversity in US collection may be due to the use of diverse genetic material from a broad geographic range and genetic pool, and outcrossing with other Pyrus species to develop the cultivars. The highest number of private alleles were seen in Asian populations, indicating historical isolations and the presence of unique genetic variations within this population. There was no private alleles in Tennessee skip populations, and there were only 2 private alleles in southern skip populations. The absence or presence of very few private alleles in skip populations show high level of gene flow within the skip populations of Pyrus scalariana. Okay, one thing, the presence of 41 multilocus genotypes in US commercial cultivars when just using 12 unique cultivars for this test indicates hybridization events as well as unintended but potential misleveling of the cultivars, misleveling of one of the cultivars as the other, because we used only 12 unique cultivars for this study. However, there were 41 unique multilocus genotypes for US commercial cultivars observed. This table shows the genetic diversity indices based on the mitochondrial SSRs. The highest gene diversity and the highest number of alleles were found to be in the locus 353. Okay. The consistency in average gene diversity between these 2 tables shows the reliability of mitochondrial SSRs in identifying genetic diversity within the PC populations. We inferred population structure using the software structure itself. Structure indicated 2 genetically distinct clusters as optimal by Ivanov's methods. The Asian populations and the TN escape populations formed 1 cluster, whereas the southern escape populations and the US commercial cultivars formed the another clusters. To see substructure by considering 3 genetic pools, we again visualized. And while keeping the 3 genetic pools, we were able to see again southern escape populations and US commercial cultivars in 1 cluster. However, Asian populations and Tennessee escape populations form 2 separate clusters. There was admixture in all of the 4 populations. However, there was a high admixture within the in between the southern escape populations and the US commercial cultivars, which indicates ongoing gene flow between the escape populations and the cultivated populations of Pyrus scalariana. We again infer population structure using DAPC. The result of the APC were quite similar to the structure. However, the clustering of agent samples was completely separate. The agent samples were the original samples. So the APC reflects the diversification of US populations from the agent samples. Here, the US commercial cultivars seem to be the intermediary between the agent samples and the escaped populations of Pyrus scalariana. So all in all, from project 2, we were able to develop mitochondrial genomic resource of Pyrus scalariana. We were able to develop mitochondrial SSRs and assess genetic diversity and population structure utilizing those SSRs. We observed overall high genetic diversity of Pyrus scalariana populations. Um, Again, the high genetic diversity among Parascalariana cultivars could be due to highly likely unintended mislabeling of the US released cultivars. So the mislabeling could have led to a mix of genetically distinct plants being grouped under the same cultivar's name. Despite the valuable insights gained from mitochondrial SSRs, these SSRs cover only a small fraction of the genome and may not capture all the relevant genetic variations. So this study underscores a need for broader genomic studies to further inform the genetic architectures of cultivated Pyroscalariana compositions. Also, we highlight the need to develop robust method for cultivar pruning and invasion management. So in future, the population genomics study can help identify the genetic markers associated with the invasive traits of Pyrus scalariana. Uh, uh, Apart from my main projects for my thesis, I also have been working with other projects related to Pyrus scalariana and other species. The one of the projects is SFRA project, which is related to population genomics of calorie pear. For this, we did library preparation and have got a tons of markers, which are still to analyze. We have also sent Bradford pear for high-C sequencing. We haven't got the results yet, but hopefully we'll get the results soon and get some insights into the population genomics of calorie pear. I also worked with undergrads, Kyle and Patrick. Kyle's project was related to diversity study of dandelion, utilizing mitochondrial and plastidial SSRs. And Patrick is new to the lab, and he will be working in SNBs in Discula destructiva pathogens. It's time for acknowledgements. Let me start with thanking USDF Forest Service Division for the arrangements of fund. A very big thank you to my graduate committee members, especially Dr. Noviki. Thank you for being the best mentor I could have ever asked for. Thank you to everyone from my graduate committee members, Dr. Bill, Dr. Ajenta, Dr. Coyle. Thank you, Dr. Tritiano, for stepping up and uh, willing to be a part of my defense on behalf of Dr. Zenta. Thanks to everyone from Dr. Noviki's lab. Said thank you for being my helpful room lab mate. Thank you to Dr. Jess Hatson from Clemson University for arranging few of the samples of calorie pear, and all the Herbaria and Arboretum for arrangements of samples. Again, Jackson, thank you again for your help. Thanks to everyone from Dr. Tritiano's lab, Dr. Harjia Brick's lab. Thank you, Meher. Thank you, Sarah. Thank you, UTIA, and our very own EPP. Thank you, everyone.
Thank you, audience, for your patience, but I hope it was worth your time, all these extra new exciting results. Time for questions. Shade has a question.
You know if Tennessee plans to start restricting sale of Iris Kelleriana? I didn't see it off the list of states.
So, Sid's question was if Tennessee plans to restrict the sales of Pyrus scalariana. There were no reports of Tennessee planning to do so, but they have been doing buyback events, like where they are trying to encourage people to try not to plant the Pyrus scalariana. Instead, they have been supplying with the seedlings or plantlets of the native species, but I have not found anything related to the plan of restriction.
What would be the value in restricting it? It's like locking the barn door after the horse is gone.
So
I
Dr.
mean, it's everywhere in Tennessee.
Yeah, Dr. Trujano asked what's the value of restricting. So yeah, it's being invasive and it's outcompeting every native species that we have. So we don't have any other options rather than restricting to prevent the ongoing spread. So it is very hard once it gets established, and it's very difficult to destroy, as I have already mentioned. So yeah, it's one of the many steps to prevent its further spread. I hope I answered your questions, or if you are not
Well,
clear.
I see no value in restricting. So, because it's everywhere in the state. So if you restrict, let's say there's 1,000 trees in the state and you restrict the sale free.
I mean, we can
What
at
does
least
that do?
prevent some further introduction and
Not
spread.
really.
No?
Any more questions? Anybody in the Zoom have questions?
How much do the mini PCR machines cost for like?
This machine, I'm not sure. Oh, okay, it's $950. Yeah.
Surprisingly affordable.
I mean, it's really cool. It's handy. That's the good thing.
16 reactions, strips.
Yeah.
Anything in the chat?
Sure. No, I don't think we have any. No.
Okay. If there are no further questions, please join me thanking Alina for her hard work again and the presentation. And there are forms to be filled out for evaluation of her seminar, so please don't forget them. Thank you, everybody. Thank you, Zoom
Thank
Land.
you all. Thank you. Thank you, Dr. Dave. Thank you so much.
See you soon, Dave.
Thank you, everyone on the Zoom.
Hey, is there another link I need to click for the private part?
We'll be changing the room, so you'll be joining with this same link, but after some time.
Okay, so just
Yes, let's do it for 11.
same link at 11. All right, I'll see you then.
Thank you, everybody, for coming.